Welcome back to Liz Lens, where today we'll be talking about vectors, restriction enzymes, and DNA ligase. Not in relation to recombinant DNA technology, but studying them or talking about them separately. So let's get into it. Now, restriction enzymes. Now we have the restriction endonuclease and we have the exonuclease. So basically, these are enzymes that cleave the DNA at one point. Now the endonuclease cleave the DNA within the DNA. So like DNA strand is quite long and it cleaves within it. But now the exonuclease cleave the DNA at the end. The exonuclease. So basically, the function of endonuclease, which is an enzyme, is to cleave the DNA at certain points within the DNA, not at the ends of the DNA. We have the type 1 restriction endonuclease, type 2 restriction endonuclease, and type 3 restriction endonuclease. So now, how are they named? How are restriction enzymes named? Now, take for example the first one, EcoR1. Yeah, EcoR1. Now, E the first letter, represents the genus, the first letter of the genus. CO represents the first two letters of the species, and R is the strain designation, and 1 is the order of discovery. So they could be equal R2, equal R3, based on the order of how they were discovered. Now, this restriction endonuclease could be classified based on the type of cut they give and based on their recognition site. Now, for the first one, based on their cuts, based on how they cut the DNA. Now we have those that give sticky ends, and we have those that give sticky ends, and we have those that give blunt end. These are very two different restriction enzymes. A restriction enzyme that gives a blunt end won't give a sticky end. Now, coming to classification of restriction enzymes based on their recognition site. Now we have the type 1 restriction enzymes, type 2 and type 3 restriction enzymes. Now for the type 1 restriction enzymes, they have recognition sites that are 15 base pair long. So now when they recognize this site, the DNA, the restriction enzyme doesn't cleave at that point, but cleaves 1,000 base pairs from that recognition site. Now that is one characteristic of type 1 restriction enzyme. Now their recognition sites are asymmetrical and it cleaves the double strand. When it's cleaving after the 1,000 base pairs, it creates a nick in both strands, the upper and the lower strand, cutting both or splitting the double-stranded DNA. When type 1 restriction enzyme cleave, they cleave to create blunt ends, not sticky ends. And type 1 restriction enzymes also have two functions, the endonuclease function and the methylase function and methylase activity. It has the endonuclease activity and the methylase activity. Now, examples of this, we have the EcoK and the EcoB. Now, type 2 restriction enzymes. Type 2 restriction enzymes are the ones that are used in recombinant DNA technology, used in cloning. So type 2 restriction enzyme, it cleaves the DNA on both strands, giving sticky ends, while type 1 gives blunt ends. Now, type 2 restriction enzymes, their recognition sites, when they identify the recognition site, they cleave at the recognition site, not away from the recognition site. So, and the recognition sites are symmetrical. So when it's cut, it gives a kind of cohesive or complementary end that can easily be attached back. For also type 2 restriction enzyme, they require only ATP for the activity. Now for type 3, type 3 restriction enzyme cleaves the DNA or cleaves the double-stranded DNA on just one strand. So instead of cleaving the DNA to separate its bolt or separate everything, it only cleaves one of the strand, not cleaving the double strand. Now, what is required for the function of this? It requires ATP and magnesium ion. It doesn't require S-adenosyl methionine. Now, the recognition sites of type 3 restriction enzymes are 5 to 7 base pair long, and they cleave 25 to 26 base pair after that. And also their recognition sites are also asymmetrical. Now, that is it for restriction enzymes. Now, out of these, the restriction enzymes that are mostly used in recombinant DNA technology or used in cloning is the type 2 restriction enzymes, because it gives because they give complementary ends, and it cleaves at the recognition site, not away from the recognition sites like type 1 and type 2 restriction enzymes. So now we come to the DNA ligase. Now the DNA ligase is the enzyme that catalyzes the formation of phosphodiester bonds between nucleotides. So after the DNA has been cleaved open, or for some reason the DNA was cleaved open, the DNA ligase comes in to synthesize phosphodiester bonds between the nucleotide. Now DNA ligase is seen in the use of is seen during DNA replication and DNA repair, and is also used in recombinant DNA technology. Now when the DNA is being replicated, double-stranded DNA is unwounded, and now there's a leading strand and the lagging strand. The lagging strand is also called Okazaki fragment. Now the strand is synthesized in fragments. It's not synthesized whole. It's more like pieces of them are being synthesized. So now eventually the DNA ligase comes in to create phosphodiester bond to seal everything up, to make it a full strand without having spaces in between. Now the DNA ligase is also used in DNA repair. Now when mutation occurs in the DNA, the bodies or the genetic system or the body system has a way of detecting those mutations and trying to remove those mutations. So now basically that points where there was a mutation, the nucleotide there could be removed, creating a space there, and the right nucleotide is brought in, and then the DNA ligase comes in to seal the nick. So now we have a DNA now that has not mutated because the mutation was removed. Now lastly, the DNA ligase is used in recombinant DNA technology. Basically after the restriction enzymes have opened up the vectors, it makes a space for the DNA of interest to come in and then seals the nick. I've done a whole video on recombinant DNA technology where I talked about this in details. Now next and lastly, we'll be talking about vectors. Now vectors are circular double-stranded DNA that are used as vehicles to carry the DNA of interest into the host organism. I also talked about this in one of my videos of recombinant DNA technology. So they act as vehicles to carry the DNA of interest into the host organism. Now these vectors, the major thing that differentiates them is their size and the amount of DNA inserts they can take. The amount of DNA they can take in kilobase. 1 kilobase is equals to 1000 base pairs. 1000 base pairs. That's basically it. So now there are some major properties of vectors that need to be considered. Now they should be able to replicate using the host machinery. They should be easily isolated and purified from the organism. They should also have some specific markers. They should also have antibiotic resistance. Now most of the vectors have actually been sequenced, like the entire DNA or the nucleotides that make them up have been sequenced. So now the restriction sites are known and can be easily manipulated to do what is needed in recombinant DNA technology. So now, examples of these vectors, we have the plasmids, we have the bacteriophage, we have the Cosmid, we have the bacterial artificial chromosome, and we have the yeast artificial chromosome. Now, the first one, the plasmid has circular double-stranded DNA, and it's mostly used in recombinant DNA technology also. So basically, they are opened up, and the DNA of interest is placed in. Plasmids can take 6 to 10 kilobase of DNA insert. That is basically like saying 6,000 base pairs to 1 to 10,000 base pairs is what plasmids can accommodate, which is quite a lot. Next, we have the bacteriophages. Now, bacteriophage are basically viruses that infect bacteria. So now the bacteriophage, when it completes its lytic phase, it gets a particular gene of interest. Now the bacteriophage it's the tail of the bacteriophage adheres to the surface of the bacteria and then injects its DNA into it. Now when it injects its DNA into it, the DNA into it is attached or kind of becomes one with the genome of the bacteria. When it goes through a phase, new bacteriophages are synthesized within the cell. Now when these bacteriophages are synthesized within the head of the bacteriophage, as it undergoes the lytic phase, it takes up some DNA from the bacteria and then it's packaged with its own DNA into the head of the bacteriophage. Now eventually this bacteriophage will destroy that particular bacteria and leave that bacteria. Now, when they leave that bacteria, they go to another bacteria and inject their DNA into this new bacteria. Now, if the DNA they got from, or the particular DNA, or the particular gene, or the particular DNA they got from the first bacteria does not exist in the new bacteria, that kind of confers or give that new bacteria a kind of advantage or made that bacteria kind of better. So that is basically how bacteriophages are used to transfer DNA from one bacteria to another bacteria. Now, we also have the Cosmids. Now, Cosmids are kind of like a genetically engineered plasmid. So it's more like a combination of plasmid and bacteriophage. They're kind of genetically modified to form Cosmids. Now, we have the bacterial artificial chromosome and then the yeast artificial chromosome. These vectors are used when large amount of DNA insert is needed. That is way more than the plasmid or the bacteriophage could accommodate. Now, for the bacteriophage, they can accommodate 10 to 20 kilo base pair, which is like 10,000 to 20,000 base pairs that a bacteriophage can actually take. And this is because of the size of the head, of the bacteriophage. That's what limits it and gives this amount. Now, the bacterial artificial chromosome and the yeast artificial chromosome are actually used in the Human Genome Project, but with the yeast artificial chromosomes, they were a bit unstable. So, bacterial artificial chromosome was what was mostly used in the Human Genome Project. So that is basically it on restriction enzymes, the DNA ligase, and vectors. So that is it for this video. Thank you for watching. Don't forget to like and subscribe. Thank you.